Good afternoon, everybody. Today, I'd like to introduce our project to develop the novel oral cancer vaccine. Presentation summary. Immunorock was established to create the world's first oral immuno-oncology IO agent of oral cancer vaccine. Immune checkpoint inhibitor ICI dramatically changed the current cancer treatment practice. However, ICI is effective in only 20 to 30% of patients. Our vaccine can convert ICI-resistant cold tumor to ICI-sensitive hot tumor. Up to now, we completed GLP safety studies and CMC development with AMED-CISB grant. We will start phase 1 clinical study at Kobe University from next year with AMED-CISC grant to confirm the safety of the product. And next, we plan international phase 2 study from 2025 to confirm the efficacy of combination with ICI. We want an alliance with pharmaceutical company to perform the phase 2 study. First, let me introduce myself. I graduated Kobe University School of Medicine in 1992 and started the translational research for urogenital cancers from 1996 at the University of Virginia. From 2003 to 2006, I conducted the clinical study of adenoviral gene therapy for metastatic prostate cancer and received the JSGT award, and the result of this trial was featured on the cover of the journal. Since 2016, I have been a professor at the Kobe University Graduate School of Science, Technology, and Innovation, and founded the biotech startup Immunorock in 2021. The development of ICI dramatically changed the cancer treatment practice. The invention of ICI was selected the Science Breakthrough of the Year 2013, and Professors Honjo and Allison received the Nobel Prize in 2018 for their achievements of ICI. Although ICI can induce excellent and long-lasting efficacy, the ratio of responders is limited to only 20 to 30%. Therefore, it is urgently needed to develop the novel agent to improve the clinical efficacy of ICIs. To improve the efficacy of ICI, the combinations take center stage in clinical trials of ICI. The function of ICI needs a pre-existing CTL in tumor tissue. The cancer vaccine forcibly induces the DC antigen presentation and the CTL, and the ICIs can activate the CTL to attack cancer cells. Therefore, the combination of ICI and cancer vaccine is a promising strategy. It is well known that some intestinal bacteria can enhance the efficacy of ICI, such as Bifidobacterium longum and Clostridium botulicum. Our Bifidobacterium vector might augment the anti-tumor activity of ICIs. So we constructed the recombinant Bifidobacterium displaying WT1 tumor-associated antigen protein. It is the world's first oral IO agent. WT1 is ranked as the #1 tumor-associated antigen and overexpresses in most of cancers. Our WT1 oral cancer vaccine displays the full length of WT1 antigen protein, and this innovative Bifidobacterium vector can deliver the antigen to gut immunity. The WT1 oral vaccine is sampled by dendritic cells in Peyer's patch and induce CTL to attack the cancer cells. Previously, many clinical studies of WT1 CD8 short peptide vaccine were performed, but the results were disappointing. CD8 short peptide vaccine directly binds to MHC class I molecule and cannot induce strong CTL. On the other hand, our oral vaccine containing WT1 protein, including multiple CD4 and CD8 epitopes, is sampled by DCs and induce strong anti-tumor immune response. While short peptide vaccine could not induce substantial anti-tumor effect, the WT1 oral cancer vaccine successfully showed us significant anti-tumor efficacy. In a syngeneic mouse bladder cancer model, the combination of anti-PD-1 antibody and the oral cancer vaccine completely suppressed the tumor growth. In tumor infiltrating lymphocyte TIL analysis, the number of CTLs was remarkably increased and the number of regulatory T cells was decreased in the combination group. We selected the metastatic urothelial carcinoma for the target of our phase 1 clinical study. This flowchart shows the current standard of care for metastatic UC in Japan. Pembrolizumab monotherapy achieved only 20% of response rate. Combination with this oral vaccine improved this response rate. Up to now, we completed the CMC development and preclinical studies with AMED CSB program. With the CSC program, we will start the phase 1 study at Kobe, Hiroshima, and Hamamatsu universities from next year. After confirming the safety in this phase 1 study, we plan to start the international phase 2 study of combination with ICI targeting expanded indication. So we want to make an alliance with pharmaceutical company for this phase 2 study. This is our proposal of collaboration scheme. As you know, the market of IO, including ICI, is rapidly growing. Our cancer vaccine will be the world's first oral IO agent. We have acquired intellectual property as a patent strategy. We have a strong patent strategy team. Also, our company, Immunorock, is receiving a full support by Kobe University of incubation company STE and have well-experienced, brilliant staffs. Thank you for your attention.
Thank you very much for a wonderful presentation.